still get the same thing. Can you believe it?
Really?
I do. Entire screen. Let's try that. Why doesn't it do that? All right, let me go back to Safari, or I mean to system preferences.
Maybe it's not screen recording. 1 second. How to share screen Google Meet for Mac. Go the Apple menu, system settings, privacy, screen recording. That's correct. We're doing it. Privacy, security, screen recording.
Wait, system preferences under which one? There's,
for instance, privacy and security. Screen recording. That's correct.
Okay. Yeah, and it's clicked. Chrome is clicked. Zoom Us is clicked, and Microsoft Teams is clicked. So all of those Zoom, I mean, it always works with all these Zooms. I don't know why it's not working here.
So system settings, privacy and security, screen recording.
Yeah, they're all clicked. Yep.
Allow applications below, and then you mark Google Chrome.
Yeah.
Put your password.
It's all clicked.
Wait, and did you close the Chrome completely?
I did. I'm going to quit Chrome. Try to.
You have quit and reopen.
Então, a Suzanne Harber é uma professora da Universidade de Rochester, também tem um appointment no McLean Hospital, que é um dos principais hospitais da Harvard. Ela é uma das últimas grandes neuroanatomistas, pelo menos que a gente conhece, da atualidade. Por que uma neuroanatomista hoje é importante na área do que ela faz, né? Porque quando a gente pensa em imagem, nem sempre o que a gente vê na imagem corresponde ao que é real, né? Então, daqui a pouco eu continuo. Vamos ver.
Select, again, the Chrome tab should, oh no, I don't want the Chrome tab. I want a window, and I want the PowerPoint. Okay. Oh my gosh.
I can't believe.
Oh my gosh. Guys, sorry about that. Oh my gosh, that was really exhausting. Okay.
So, André, let's make a very short introduction and let's start.
No, don't even, don't worry about it. All right.
Bom, pessoal, eu queria falar um pouco com vocês, né? Hoje é um prazer enorme a gente dar boas-vindas a todos vocês, né, para esse nosso encontro, né, para esse primeiro encontro nosso científico. E a gente tem uma honra muito grande de receber a palestrante e convidada, a doutora professora Suzanne N. Harber. E o que eu queria falar para vocês é encorajar todos vocês que aproveitem essa oportunidade única de aprender com uma das maiores mentes, né, na área, como o professor Eurípides mesmo já falou. Depois da palestra, se vocês quiserem interagir, mandarem as perguntas, o professor Eurípides vai estar selecionando 2 perguntas para a gente poder estar fazendo a professora Suzanne. E eu espero contar muito com a participação de todos vocês nessa palestra hoje à noite, né? E I'm myself grateful for your presence in this evening and wish you an excellent lecture, professor. And now I pass the floor to the Professor Eurípides.
So thanks, Suzanne. I was going to tell them how grateful we are, but I don't want to spend more time doing that. Let's hear you. Go ahead, please.
Okay, thank you, everybody, and sorry about this problem. We're still having a problem here. Why can't I?
We are seeing you.
I know, but I can't start the presentation on this screen. Oh my gosh.
We are seeing the presentation.
Well, I know, but you can see the presentation, but okay. Can you see the presentation now?
Yes, you can.
Okay, can you see it now?
Yes.
Now it's working.
It's perfect.
Okay, Okay, so hi everybody. As you heard, my name is Suzanne Haber. I'm at the University of Rochester, which is in upstate New York. Everybody thinks it's in New York City, but just, you know, it's 5 hours from New York driving. And also I have a lab and a clean hospital, part of Harvard Medical School. What I'm going to talk about today is this procedure called deep brain stimulation, or DBS, for psychiatric and neurological disorders. I don't know how much you all know about it or what the goals are for this. So I'm going to kind of go through how we think about the pathways that are involved in deep brain stimulation and the work that we've been doing in that area. We mainly focus on obsessive compulsive disorder, but we also collaborate with people in depression and PTSD. So for those of you that are completely unfamiliar with it, let me just play this little video.
The brain is made up of millions of connections between neurons that control movement and behavior. Different areas of the brain have different regulatory effects on these neural networks. Disruption of the regulatory portions of these networks leads to various neurologic and psychological diseases, such as Parkinson's disease and essential tremor. Deep brain stimulation has been found to relieve the symptoms of certain of these diseases that are resistant to medications. Deep brain stimulation involves surgically inserting an electrode into the central portion of the brain in the part called the basal ganglia. The electrode is then connected to a generator similar to a cardiac pacemaker. The rapid firing of the electrode interrupts the abnormal networks, relieving symptoms such as tremor and muscle rigidity in Parkinson's disease. Before current medical therapy was available, Parkinson's disease was treated with ablative surgery, which involved the non-reversible destruction of brain tissue. With deep brain stimulation, the firing of the electrode can be adjusted in an outpatient setting in order to control the symptoms, but to minimize unwanted side effects. Currently, deep brain stimulation is approved by the FDA for the treatment of Parkinson's disease, essential tremor, and dystonia resistant to medical therapy. Ongoing research is studying the treatment of severe depression and obsessive compulsive disorder by stimulating alternative areas of the brain.
That's a little introduction to deep brain stimulation. And as was indicated in the video, movement disorders has been the first area that in the brain that has been used for deep brain stimulation. And there are particular subcortical regions that are stimulated, the subthalamic nucleus, the globus pallidus, and parts of the thalamus. Now, the difference between movement disorders and the psychiatric illnesses that are now being explored for deep brain stimulation, and over here on this side of the slide, you can see the electrodes with the 4 contacts embedded in the different regions in the internal capsule and the ventral internal capsule into the striatum and to the STN. And for depression, which I'm not going to talk about today. But one of the main differences between these two sets of disorders is that the electrodes that are placed for Parkinson's disease or the movement disorders, one can very quickly see the results because you turn the stimulator on and the tremor immediately stops. You turn the stimulator off and the tremor returns. With respect to the mental health disorders, OCD and depression, that is not what happens. It's more like the pharmacological treatments where you have to wait a bit to see whether there's an effect. You have to adjust the electrodes in the sense of the parameters. You don't move the electrodes around in the brain, but you change the bandwidth, the frequency, etc., which you can do at each of the contacts to get the most effective treatment. But you don't actually have the readout as you do in the motor disorders. And one of the other issues then that kind of raises is that you don't even know in the surgery, during the surgery, or even after the surgery, whether you're actually in the right place, because there's no way to test it. Whereas in the movement disorders, as I mentioned, you can see right away whether you're in the right place by turning the stimulator on and off. There's a lot of electrophysiology that's often done during the surgery, again, to assess movement. But psychiatric illnesses, again, as I mentioned, are more difficult to assess the best area. So that's a little overview of the DBS approach, and we'll come back to that. Now, the main areas that are being affected in deep brain stimulation are the white matter, particularly the myelinated fibers. And even though I'm going to be talking about DBS, TMS, or transcranial magnetic stimulation is also, the target is also the white matter. So it becomes very important for us to understand how those white matter fibers are coming through the internal capsule or whatever fiber bundle you're targeting in terms of what fibers are traveling where. And this is sort of a schematic, but from the diffusion MRI literature showing the funneling of fibers down into the internal capsule. So as I mentioned, the myelinated fibers are preferentially stimulated over the non-myelinated fibers. Axons are more easily stimulated than cells. And then the electrode, as I mentioned, are all often preferentially put in myelinated fibers. Here's an example of the electrodes. There are 4 contacts. As you target a particular brain region, you have the option of stimulating any of the 4 or a combination of those 4. So this introduction is to kind of emphasize how important it is for us to understand the white matter connections, and particularly for DBS and for other treatments, such as TMS. And so this area of white matter connectivity has kind of got lost a little bit in the literature as people had been focused more on brain regions as opposed to the white matter, the connectivity. But that has actually changed in the last few years with the addition of diffusion MRI as a method to follow those pathways. But of course, lesions and deep brain stimulation is new, but lesions for psychiatry is not new. And so there were, very large lesions. Basically, most of them are focused on disconnecting what's called the limbic system or the emotional system in psychiatric illnesses. And they're, kind of got a bad rap, the frontal lobotomies, where large areas of brain were disconnected, again, focusing on the prefrontal areas. There was orbital undercutting, again, trying to address orbital fibers, an area that's always been associated with psychiatric illnesses. The subcaudate tractometry, which is not that different from the orbital undercutting. There was anterior capsulotomy, which is actually still done today as instead of deep brain stimulation, but with much more elegant and advanced techniques. So the lesions are small, but those were also effective. And then there's cingulotomies, an area of lesioning the cingulate cortex. And both of these 2 techniques are actually used still today quite effectively. But again, the lesions themselves are relatively small. So there's a long history of trying to disconnect white matter connections to help psychiatric illnesses. And so one of our main interests is what are the white matter pathways that are actually passing through. And I'm going to focus a little bit today on the internal capsule. That's going to be our main focus because it is a target for DBS. But what I'm going to tell you in terms of concept is going to be true for whatever area of brain you're targeting for either deep brain stimulation or even TMS or lesions. So the questions we're asking is how do fibers, how are they organized and how do they go through the internal capsule? And then how well does diffusion MRI identify this organization? And as you can see, for a very long time, we've had a very good map, a general map of the internal capsule as depicted over here on the left. And I'm sure all of you have seen that in your anatomy textbooks where various things come through the internal capsule. But if you actually look at this very closely, you'll see they're mostly motor systems or premotor systems or sensory systems, right? So then if you look at how people think about the internal capsule, we have the motor, premotor, and then all of the prefrontal cortex, okay? But you may not want to target, like you did in the frontal lobotomies, the entire frontal cortex. We have much better ideas about the symptoms that are, that we understand the symptoms better in illnesses and how they parse out to the different kinds of illnesses or subpopulations of patients within an illness. And we think about those in terms of the cortical areas that may be dysfunctional. So having this as one great group of fibers may not serve our purpose as well anymore in terms of trying to target specific connections and getting away from those large lesion-like approaches. We don't use the large lesions like the frontal lobotomy, but trying to be a little bit more precise to what we want to do. So there's a long history of anatomy, which I just showed you a quick slide of. And even though that was the origin for our understanding the brain, of course, imaging studies have now taken the lead in understanding psychiatric illnesses, linking behaviors to circuits, and understanding circuit dysfunction. And on this side, we have some functional MRI, where a number of different networks have been identified over time. And I'm not going to go through this because that's not really the point of this particular talk today, but it is a connectivity profile that people have been addressing and looking at. On the right-hand side, we have the diffusion MRI, and the diffusion system and the way these are actually following these fibers is, of course, it's an indirect way, and it follows it's organized along how the water molecules are oriented within the tissue, and the lack of, the more oriented they are, the higher myelinated areas there are. And so it really is based quite a bit on the orientation of myelin. So one of the things that is very important is that these methods, both the MRI, the functional MRI, and the diffusion, are indirect measures of connectivity. So we don't really know what underlies those measures other than the water orientation, but it doesn't actually tell you the hardwiring of the axons clearly. And so for this question, the gold standard still is the non-human primate, and we use the non-human primate because the frontal cortex is so much more advanced compared to rodents and has a lot of homologies that have been identified between the non-human primate and the rodent, and the human. So the challenge really is, sorry, you can still see my screen, or what did I do? It's still okay?
Yes.
Yes.
Okay, perfect. So the challenge then is to take the gold standard, as we know it in non-human primates, and translate what we know about those connectivity where you see the axon under the microscope, it's an axon, you don't know it, you don't think it's sort of water molecules that may or may not be an axon. And how do you do that? How do you take those that data and actually, I have to go back to the other screen there. How do you actually take that data and understand the human brain from that? So the question is, how do we use the precision of anatomy based on animal studies to inform targeting? And in general, the first question we asked in this particular case for the internal capsule is, can we segment the white matter bundles based on the cortical connections traveling through those bundles? I showed you a slide before that segmented in terms of motor system, but we really want to segment it better and identify the various prefrontal areas and how they are organized within the capsule. So the question is, how are they organized? And we're, of course, using non-human primate data for that. How well does the diffusion MRI demonstrate this organization. And then we'd first do that in the monkey and then in the human. And then how can we use that information to better target for DBS? So just as an overview, we do our experiments in the non-human primate. These are tracer experiments, which I will explain in a moment. We sacrifice those animals. Those animals are then shipped to Martinez Center at Mass General, where high resolution diffusion is done in the same animal that we have the tracer injection. And then we can register those together to see whether or not the diffusion that we're seeing in those animals after we place a seed in the area where the injection site was actually represents the real fibers that are in that brain. From there, once we understand the diffusion a little better and we understand what's real and what isn't real in the diffusion, we then move to the non-human, to the human primate, and look at just the diffusion, because we can't do tracers in the human, of course, and try to apply the same rules that we see in the monkey tracing to the human. And from there, we can evaluate various diseases and look at the abnormalities within those white matter tracts, and then start to think about what areas and how we should target for the deep brain stimulation. So what we're going to do is talk about defining rules for those axons that from specific organization within the bundles, and how different areas travel, what position do they occupy in the bundle. Again, as I said, can we see this in the diffusion MRI, first in the monkeys, then in the human, and can we use this for targeting? So just as a review, I know you've all looked at cortical maps before, especially in medical school. There's an orbital view of the human and a sagittal view. I don't have a lateral view right now, but in this figure. But here's the monkey below it, and the homologies have been worked out very well for many, many years. And here's some of the several groups that have worked on this, and they really have come to very similar conclusions. So what that means is if we're talking about, say, anterior cingulate or area 10, and we're following fibers through there, we can find the same area across species and ask the question, are they doing the same kind of thing? And we know that the kinds of trajectories they have to take are going to be very similar, because they have to go through similar cortical and subcortical regions to get to their target. This is just kind of an overview that we know the different areas, and I don't want to go through these in detail. It's a snapshot of what we think these cortical regions do, and it's based on lots and lots of literature in the human functional imaging world, and in wake behavior monkey physiology. And so we have a lot of good ideas about what these different areas do. And so what we want to know is, okay, we have these functional distinctions to some extent. How do those areas, how do the fibers from these regions go through the internal capsule? Because you may, your hypothesis may be, for example, that which is one of ours right now, and in terms of OCD, that attentional switching or behavioral flexibility is a problem. And so maybe if we target the ventrolateral prefrontal cortex fibers in the capsule, that may help in the therapy. Likewise, the dorsal anterior cingulate is another area that there's a lot of evidence that's involved in OCD and in depression. And so the question is, can we find where those fibers are going through the capsule, and can we identify those to target better our leads? The striatum is organized in a very similar way, with a kind of a limbic, cognitive, and motor system area. And the targets for deep brain stimulation will be in both. Here are our injections on the lateral, orbital, and medial. You can see we have many, many injections that we're going to follow. Okay, so let's get back into what we're actually doing. So what are tracers and transport? The axonal transport, the tracers use active axonal transport. These have been used for a long time. I still have modern on here, and the reason I have modern is because many times in these lectures, I go through some of the very old methods that we use to understand anatomy. But these aren't the modern ones anymore, at least in rodents. There's a lot more new ones, but they're not that effective in non-human primates. So we still use these at the anatomic, the axonal tracer molecules that use active tracers. And so what you do is that there are two kinds. They're anterior grade, and here you're injecting a tracer in an area, and the cell bodies in that region pick up the tracer, and it's transported to the terminal field. The other direction is also used. You can inject it in the terminal field, and you label the cell body. So in both cases, or many of the tracers transport in both directions, from the point in which you inject the tracer, you get the full perspective of where they project in the brain and who's coming to them, who's projecting to them. So you get the cells and the terminal fields, and of course you get the fibers. And this is the part we're most interested in. I mean, in this lecture, that is to say. Okay, so here's an example. Here's an injection. Here are the fibers coming up. You don't even need a microscope. They're very beautiful. They're just kind of, and here they are. Here it is in dark field and blown up. You can see that these are clearly fibers. There's no question about it. You can see the individual fibers on the side. I blow it up even more. And these are really nice illustration of how we can trace these. And at the same time, you have the terminals that I mentioned, and you can label cell bodies. We're going to, again, focus on the fibers, not the cell bodies, but just to let you know, these tracers really are very effective in giving a complete picture of the connection of these brain regions. And so then what we do is we trace these out laboriously under a microscope, and then we render them in 3D. And in this top left-hand figure, you can see the 3D organization of the fiber bundle. Then I think actually this was an injection in the medial, ventral medial part of the cortex. And you can see this is the entire fiber system of that one going up into the corpus callosum, et cetera, and you can see that it doesn't go throughout the whole brain. You also see the cells over here, which again, we're not going to talk about. And then what we can do is take all of the injections together and look at their relationship to each other. And again, this is just to remind you, if you had an electrode coming in somewhere, you would want to, and you have 4 contacts, and I'll show you pictures of it later, you want to know where these fibers are vis-a-vis the other ones to know which fiber bundle you're actually going to. And here's just a little movie showing you one example of what we do. Here's the injection site, and they're going to go away. And now you can see these are 3 other injection sites. You can see they all go into the uncinate fasciculus. They're coming over to the corpus callosum, up to the cingulum bundle. And you can see the relationship between these 3 areas that have different positions in the brain. And now they're going in the internal capsule, our area of interest, and those fibers are extending into the thalamus and down into the brainstem. And so you can look at the internal capsule, and you can see that these 3 injection sites, here's the anterior commissure for some orientation. You can see that they are organized in a very, very specific way. So one of the targets, of course, is the ventral striatum. So I'm just going to have one picture showing the organization of fibers in the ventral striatum, which is somewhat topographic. But if you put them all together, again, in this ventral striatum, unlike what I showed you in the internal capsule, there's a great deal of mixing, although there's a general topography. And you can see the mixing here on these 3D renderings. And again, we'll come back to this figure when we see an electrode into this region that's trying to target maybe the ventral striatum or maybe the internal capsule. We mentioned the subthalamic nucleus, the ZI. Don't pay attention to that. We're not going to talk about it. But we mentioned the STN. It's a target for Parkinson's disease, but the medial part of it is also a target for OCD, and an effective one. It's more used in Europe than in the US, but people are successful in it. Again, a similar situation as with the striatum. There's a general topography, but a lot of overlap between limbic and cognitive, and in this case, even some of the premotor areas. Again, we'll talk about the electrodes that go into those regions. So, yeah, this is just a summary that these areas converge, many areas converge in the rostrostriatum and the rostromedial STN. But I want to move on to the internal capsule because that's how these guys are getting their fibers from, and want to ask the question of how they're organized within the capsule. So the first thing that happens is what you're seeing here is that there's an injection site in various places along the coronal section of the brain, and the fibers come into this general white matter region. And what's important here, and the reason we follow them not just in the capsule, but we have to trace them completely from their area of origin, because how they come into these regions will dictate how they get into the capsule, and then that will dictate their position within the capsule. So the first thing we do is we follow these fibers, and they, no matter where they're coming from, there's this tight bundle. This is the histology. And within that tight bundle, there's 3 sets of fibers that are going to go in different positions. So if you're coming from the dorsal, this is coming the dorsal area, one set of fibers will peel off and go medially, and they will go into the corpus callosum and the cingulum bundle. Another set of fibers will peel off laterally and go into the capsules, the internal, external, extreme capsules, and one will come down and go into the striatum. And the same is going to be true if you're coming from the ventral surface or from the lateral surface. And what we found as we get into the internal capsule is there's a very clear organization as fibers come into the internal capsule. And again, what was very helpful is understanding how they enter the capsule. There's this very clear dorsal ventral topography. So the dorsal prefrontal cortex fibers are dorsal to those from the ventrolateral, which are dorsal to the OFC as they go through. And you can see that they pretty much take up the capsule because, of course, you can imagine more fibers from different areas. I'm only illustrating 3 are coming in from the prefrontal cortex. But as you move caudally now, some of the premotor areas and the motor areas and these more varicaudal areas are going to come in, and they push these fibers more ventrally. But they still maintain their topography. And below, you're seeing the histology of how that actually looks. So this is another little film that shows many, many different injections throughout cortex and their trajectory to the internal capsule. That's the motor system, pretty much straightforward down. The premotor, and what you can see right away is that they have a very straight trajectory, not very complicated. But the ones from prefrontal cortex start to get wiggly and squirmy because they have to get into the capsule. They have many things to do. They have to go caudal, they have to go ventral. And so each of these have their own trajectory to get into the capsule. And so we've just put a bunch on here. So you can see that it's complicated, but through the complication, you can see that there's some organization to that. And you can also see, as we'll go through later, as you, let's see if I can get this back to here. If you put an electrode in, again, you can see that you can preferentially target some groups versus others. So based on that, we demonstrated that there's this medial-lateral organization and dorsal-ventral organization. And so the capsule looks a little bit like this, with this kind of organization within it. Then we wanted to know how they get down to the thalamus, the STN, and the brainstem. And we followed the fibers, and here you can see they start to turn as they go into the thalamus, and they enter the thalamus. They continue to go. These, again, prefrontal cortex fibers. They enter this area called the zona incerta. They continue down. They peel off to this STN, and they continue down to the STN, to the brainstem. And so we haven't charted all of those all the way down. We're in the process of doing that. But what's important then is to see where each of these set of fibers from different cortical regions peel off and enter these different regions. And what you can see right away is that the motor cortex just comes straight down. And so electrode anywhere over here is just not going to affect the motor system. And likewise, when you're targeting the motor system, you're unlikely to affect the other areas that much. Okay, so then the question is, how well does the diffusion show that? So remember, I mentioned that we're doing diffusion in the same animal that we do the tracer in. And so here's an animal that has an injection right here. It's the tracer. You see the beautiful fibers coming up. You can't see it very well, but they're crossing the corpus callosum, going through the white matter. And here's the exact same animal registered for its diffusion MRI. Here's in blue the injection site, and very beautifully follows the same organization. So this was really very exciting to see, but of course it's no surprise to know that there are also a lot of false positives. There are also false negatives, but I'm going to talk about the false positive. In the yellow arrow, you can see the fibers coming in. They're coursing around the stratum and will enter the internal capsule. That's the correct way to go. Obviously, it's what the histology is telling us. And here in the diffusion, you can see that there is a correct one. Here, these fibers, they haven't entered yet. They're just turning around. This is what this is doing. But there's an incorrect one where this blue arrow is, and they're dripping in, and that's not a correct one. And similarly, you see in this more caudal section where the fornix is labeled in the diffusion, but there's not any fibers. So then we ask the question, okay, this problem of getting into the internal capsule, we see it, but we also get a very big false positive. What happens in the human? So this is what you just saw in the monkey histology and the monkey diffusion. And yes, exactly what happens in the monkey diffusion happens in the human diffusion. So we have the correct one, which circled around in a more rostral section and entered the capsule in the correct place. And then you have this big blob coming in, which is incorrect. And this is a little bit of a turn view so that you can see how it's correctly entering. It's correctly entering the internal capsule. So based on this, and this is one example of many, many different cases, we showed that the same kind of rules of getting into the capsule can be shown both in the monkey and in the human. And here's, again, the rule of the dorsal, the ventral organization. This is from our 3D model, the movies that you saw. Here are the injection sites. Here they are in the capsule, nicely dorsal and ventral. And here the same is in the monkey diffusion. Again, you see the same kind of organization. And then in the human, when we place the seeds in comparable areas, again, you see the same kind of organization in the internal capsule. What's really important, though, is that there is individual variability. And here we then took the rules that we identified and asked the question, how does that look in the individual cases? And here are 3 out of the 32 cases, individual cases that we tested it in. And what you can see that is that, for example, this ventral medial prefrontal cortical red region is very ventral here. It's not so ventral here. It's not very ventral here. Oops, I'm sorry, it's the wrong one. That's the medial. That's the lateral surface. But there's a different position in these. But the important point is that, and we expected them not to be a point to point, but what's important is the topology is maintained. So the dorsal one's always dorsal, mid, ventral, and so on, and we see that throughout. And the same is true on the lateral surface, just showing you that's not just the medial, but the lateral surface shows the same rules. And we did that with the medial and lateral organization of the internal capsule as well. And this is showing that exactly that for example, the lateral areas like dorsolateral prefrontal cortex, as opposed to the anterior cingulate, you'll have a medial lateral organization. So, so the same kind of organization that I just showed you in the monkey, we can now fairly confidently see is true also in the human. So what about going down to the brainstem? The brainstem, we have a very high resolution scan of an individual, and here it's not one that it's so high resolution the individual was in the scanner a very long time, because we're really looking at very, very fine fiber trajectories that are actually peeling off the internal capsule. So your clinical scanner isn't going to pick this up. But we want to make sure, though, or we wanted to ask the question, is the same organization that we saw in the monkeys true in the human? And indeed it was. If you recall, as you get close to the thalamus, the fibers change orientation in the histology, and they do that in the diffusion. As they get to the ZI, they peel off, and the others continue down, with another portion coming into the STN, which is happening here and starting here, and then the final one going down to the brainstem. So in this way, we really can show that a very similar kind of organization is true. And we also showed that in this case, the green was the prefrontal areas, and the blue ones were the more motor areas. And you can see the green going into the thalamus first. Okay, so what does that mean for deep brain stimulation? Again, I just want to remind you that the movement disorders situation is much easier, because you can really test whether the deep brain, the stimulator is in the right place, right in the surgery room, and that's not true for the mental health ones. So we really want to have a hypothesis for the mental health ones: the ventral internal capsule, striatum, which is one target, the striatum, not really one, technically two, and the STN, which I don't have here. But so we want to understand where those fibers are going. Again, a reminder of the cortical areas that one might hypothesize in different situations that might be involved in these different areas of cortex, and ones that you might want to target. Okay, again, I'm reminding you of this topography. So here are the leads. So there's, for OCD, there's the internal capsule, the anterior limb of the internal capsule, in particular, the ventral striatum and the STN. These two are pretty much the same target, but there's been back and forth between what you call it. But in any event, they're very similar in terms of their trajectory. And so here's one these are old, again, images. People have moved these targets a little bit, but this electrode with its 4 contacts is in the ventral striatum. This is in the internal capsule, and this is in the STN. And so, you can ask many questions, which we won't really go through a lot of them, but the question we're going to talk about now is which fibers pass through each site. Another very interesting topic is what do they have in common and how do they differ, which we've thought about as well, but we won't be able to go through that here. So as I mentioned, I was going to put an electrode in the picture of the striatum you saw. And so here's the ventral system with this sort of mix of orbital and ventral medial prefrontal cortical areas and the ventral anterior cingulate cortex. And then in this area, you have a little bit still of the dorsal anterior cingulate, it's a little bit mixed in. Here we pretty much are moving out of those very ventral systems where you have the anterior cingulate, but we're starting to get a little bit of the dorsal lateral and dorsal medial and the ventrolateral prefrontal cortex. And you keep coming up and you get more and more of these different fibers coming through, but fewer of this sort of very, very clear, classically limbic areas. And up here, you're starting to get the motor system coming in, and they're not part of the target. The interesting thing about this is that one of the things that researchers have to pay a lot of attention to is that there are other things going on here. So you have an electrode that's going through a white matter track, and you're thinking you're in the striatum with all the striatal fibers, but of course there are a lot of things, cortical fibers, but there are a lot of other things going through there. And one of the things I want to highlight is, for example, when several groups started doing it, they wanted to get these limbic areas, and so the electrode was deeper. And you did get a lot of these regions that they wanted to. But sometimes the patients had a lot of anxiety, and it really was the opposite of helpful to the patient. And it's not just that these areas are very limbic and visceral to some extent, but this is also the area that the amygdalofugal fibers come through. You're very close to hypothalamic fibers. So there's a lot of gut stuff going through this very ventral part of the striatum. As a result of the clinical outcome, and in part because of what we were working on, they figured, okay, well, the electrode up here is going to be, maybe a little bit more out of that region and maybe more into the dorsal anterior cingulate and non-visceral kinds of regions. And so the electrode was moved a little bit more dorsally and a little bit more caudally. So here's just a schematic of, again, as you can imagine, you can put the electrode in, and this again is an old slide and not color-coded because it just isn't color-coded. But you can see that if you're down in the VMP, if you're catching VMPF fibers, it's the first contact. Orbital is a little bit more spread out. The dorsal anterior cingulate is a little bit more dorsal. And for the most part, your dorsal prefrontal cortex in the old system was pretty much outside of most of them. Maybe contact 4 would get some of the fibers captured here. As I mentioned, the idea now is to be a little bit more dorsal. The STN is a similar case in which the medial STN, lots of convergence. But again, wanting to point out that the fibers that people hypothesize they want to get are the fibers from the cortex, the hyperdirect pathway, and basal ganglion fibers. But again, in pointing out this is a very, very white matter rich area with lots of stuff coming in and out. That is not necessarily, it still has an effective outcome, but the point is that if you're trying to link your outcome to an actual cortical or particular brain region, it's important to take that into account. So in summary then, the cortical fibers can be segmented pathways within the internal capsule. Anatomically, we can link the tracing to the diffusion MRI, first in the monkeys and in humans. And we found that to be a very important step in interpreting connectivity and the specific pathways for humans in the internal capsule. And that segmentation can guide us to areas that are good to target for both invasive and non-invasive therapeutic approaches. Obviously, didn't have time to talk to non-invasive ones. And segmentation is also very important for individualized targeting. And I think I'm going to end here with thanking many, many people that have worked on these projects. Our Conti group, which is part of our large group on OCD, and many of the people in the lab. And with that, I will stop sharing. I will end the show. So that's it.
Thanks a lot, Suzanne.
That's terrific.
Thanks a lot for your excellent talk. I will make a few comments, Suzanne, and if you don't mind, I will say in Portuguese because I think it could be.
Oh yeah, of course.
And then I will make 2 questions.
Okay, if I stop presenting,
Please stop sharing.
I won't go away. Okay, good, I'm not away. Okay, good.
Então, boa noite a todos. Estou muito feliz de estar aqui com vocês. Essa aqui é mais uma das atividades que envolve o CISM, nesse Centro de Pesquisa e Inovação em Saúde Mental, que tem uma parceria tanto com a Unifage, com a Unimax, quanto com a USP, Universidade Federal do Rio de Janeiro, do Rio Grande do Sul, e a Unifesp. Então, é muito gostoso para mim estar aqui com vocês, interagindo e trazendo para vocês uma pessoa que a gente admira muito. Eu resumi um pouquinho, bem rapidamente, como é que eu vejo essa área da Suzane, como é que eu resumiria a aula dela para vocês. Eu diria para vocês que nós estamos falando de uma área de interface entre várias disciplinas. Então, aqueles que querem fazer medicina, neurologia, psiquiatria vão ter aqui uma área de atuação, aqueles que querem fazer neuropsicologia, os psicólogos, e entender mecanismos cerebrais, também vão encontrar nessa aula uma porção de fonte de inspiração. Aqueles que um dia querem estudar matemática ou física vão encontrar aqui uma porção de áreas, porque muito dessas imagens são produtos de algoritmos matemáticos. Então, há uma área de interface de inúmeras profissões. Mas ajuda vocês também a ter uma visão de perspectiva futura da psiquiatria e da neurologia, né? Então, esse tema que nós estamos falando aqui envolve neurocirurgia. Neurocirurgia a gente só usa em transtornos mentais e neurológicos que a gente não resolveu de nenhuma outra forma. Então, no caso da psiquiatria, as áreas mais estudadas é o transtorno obsessivo-compulsivo e a depressão. Aqui ela focou mais no transtorno obsessivo-compulsivo. A ideia é que esse transtorno obsessivo-compulsivo, que envolve ter pensamentos que vêm à mente do indivíduo, que incomodam profundamente, trazem muito desconforto, estão associados com o desconforto, para aliviar, o indivíduo se engaja em comportamentos repetitivos, por exemplo, medo de se contaminar e lavar a mão diversas vezes para aliviar, mesmo ele percebendo que aquilo é exagerado, que não faz sentido. Você acredita, você observou que você desconectando certas áreas cerebrais, você melhora profundamente esses pacientes. Eu estou falando de pacientes muito graves. Uns 50% deles melhora muito com as intervenções neurocirúrgicas. Entre as intervenções neurocirúrgicas tem a ablação, que é você lesar, desconectar áreas cerebrais, e outras são essa neuromodulação, que vamos dizer, é menos invasiva, menos agressiva, você coloca o eletrodo lá, e você pode estimular conexões cerebrais. A ideia é que os transtornos mentais, eles refletem alterações de conexões cerebrais, conexões entre áreas do cérebro. Então, se você inibir certas conexões cerebrais ou estimular certas conexões cerebrais, você pode melhorar. E o fato é que melhora profundamente. O campo está evoluindo no sentido de cada vez você fazer lesões mais menores e mais localizadas ou estimular áreas com maior precisão. Só que, para você estimular áreas com maior precisão, você precisa saber qual que é a área que é importante, ou seja, saber mecanismo de doença. Mecanismo a partir do qual essas conexões, esse funcionamento cerebral está alterado. Então, você tem que usar métodos como de neuroimagem, como esse diffusion, que ela fala de Fusion Tensor Image, né? Imagem de difusão, que estuda a conexão cerebral, esses circuitos cerebrais, que você vai querer, no toque, então, esse transtorno que eu falei para vocês, há uma alteração nesse circuito córtico estriato-talo-cortical, esse circuito X, e eu vou identificar que ele está alterado naquele indivíduo e eu vou colocar um eletrodo lá e estimular aquela região cerebral. Só que esses métodos de imagem, que são a ressonância magnética, eles são métodos que dão uma medida indireta do nosso cérebro, não uma medida direta. Ela usa, de novo, algoritmos matemáticos ou propriedades da difusão da água. São medidas arbitrárias e indiretas que correspondem com um certo grau de realidade, mas com um certo grau de incerteza. Ela usa, então, macacos, primatas, não humanos, né? Primatas, e usa uma técnica que ela é a maior autoridade na área, que coloca um traçador, ela coloca numa área específica do cérebro, um traçador que tem a propriedade de trafegar, né, de ser transportado pelo axônio do neurônio até o corpo celular, ou de uma forma retrógrada, do corpo celular para o axônio. Mas, com isso, ela tem uma idade muito mais fidedigna de quais áreas do cérebro estão se conectando com quais áreas do cérebro. E, embora os primatas, os não humanos, tenham um cérebro um pouco diferente do nosso, as regras a partir dos quais, evolutivamente, essas conexões cerebrais se desenvolveram são mais ou menos, se repetem em nós humanos. Então, ela estuda e demonstra quais regras esses circuitos cerebrais, essas projeções cerebrais seguem de indivíduo para indivíduo. E daí, ela então estuda o primata, vê lá, tem que, para você observar, usar esse método termina com o sacrifício do animal, e você vai no microscópio e vai ver, no microscópio, como é que essas leis ocorrem, como é que essas conexões se estabelecem. Então, você tem que matar o animal, então não dá para fazer isso com humanos. Então, ela vai lá, pega o animal, estuda essas vias, faz uma ressonância magnética utilizando esses métodos de difusão que a gente usa em humanos e começa a ver quando as limitações do método, os falsos positivos, os falsos negativos. Com isso, ela pode ajudar a melhorar os métodos, mas ela pode ajudar a gente a fazer essa tradução do método em primatas para os humanos, se a gente, então, aprender a localizar com mais precisão como é que esse circuito se organiza no cérebro. Então, uma das áreas que ela estudou muito é essa área da cápsula interna, né? Uma área de projeção do córtex para os gânglios da base, ali entre o núcleo caudal e o núcleo putâmen, né? Também não precisamos entrar em detalhe de anatomia agora, mas você tem uma área grande ali de projeções dessas estruturas de várias áreas do córtex pré-frontal, e a gente já foi aprendendo nos nossos estudos quais áreas são mais importantes para lesarmos ou para colocar o eletrodo e estimular. Eu acho que essa é uma área de futuro e muito interessante para vocês, porque nós estamos falando de métodos muito invasivos, neurocirurgia. Mas qual que é o grande perspectiva que esses estudos trazem? Que uma vez que a gente conhecer essas vias, a gente pode então usar métodos menos invasivos para estimular ou inibir essas vias. Uma vez que a gente sabe que fazem parte do mecanismo da doença, a gente pode intervir na doença, intervindo nessas vias, utilizando métodos que são menos invasivos. Então, a gente tem hoje a estimulação magnética transcraniana, não sei se vocês já ouviram falar, a estimulação direct current, direct current estimulação por correntes diretas, que é elétrica. Agora tem uma outra chamada low full, ultrassom de baixa intensidade, que vai conseguir interferir ou modular vias mais profundas do cérebro. Então, eu vejo nesse trabalho da professora Suzane uma área de grande interesse e perspectiva futura, e eu acho que é legal vocês estarem atentos e vendo o que está acontecendo nessa área tão interessante. Então, se vocês não se incomodaram, eu vou fazer 2 perguntas aqui, bastante interessantes, né? So, Suzanne, may I ask you 2 questions? So I'm going to translate it to English. Given the complexity of interactions between the prefrontal cortex and the basal ganglia in relation to decision-making and reward-based learning, how do you see the evolution of DBS techniques in terms of personalizing treatments for specific dysfunctions in these circuits? Is there a potential for development of adaptive algorithms that can adjust DBS parameters in real time based on patients' neural activity?
Wow. Okay. That's a very complicated question. Can you read that again? Sorry, can you just...
I think the first part will be interesting. Given the complexity of interactions between prefrontal cortex and the basal ganglia in the relation to decision-making and reward-based learning, how do you see the evolution of DBS techniques in terms of personalizing treatment for specific dysfunctions in these circuits? Maybe you could tell us a little bit about your model of persistent avoidance and the ventrolateral prefrontal cortex as part of this circuit involved in OCD.
Yeah, so I think there's several questions in there, and there's several, I mean, you know, there's a lot in there, packed in there. The relationship between cortical basal ganglia circuits and what they're doing is a really, that's a whole text by itself. So one of our, what Yuri is referring to is that one of our hypotheses is that as we look at the brain, at the different areas of the brain that, I mentioned, we tend to have these sound bites of function that are associated with them. But a lot of what we're thinking about in OCD is this kind of an imbalance, if you will, or in the ability to be behaviorally flexible, right? So in most of the world, in your daily lives, you balance between getting a reward or a positive outcome to an event with the negative possibilities of that outcome. And obviously, if there's a great reward and very little negativity, most people will go for that reward. But we're constantly making those decisions. And one of the hypotheses we have right now in OCD is that balance, which is a combination really of cortical, and cortical subcortical circuits. So getting to both the basal ganglia and some of the brainstem regions or deeper regions, the thalamus. It's very hard to know the role that everything plays, but the balance between reward and negative outcomes, positive versus negative outcomes, is really an important one and allows us in our daily lives to be pretty flexible depending on what was learned, previously, just 2 days ago, yesterday, right now, or a few years ago. You kind of balance all of those things and you change your behaviors accordingly. And one of our hypotheses is this behavioral flexibility is lacking in or disrupted in OCD patients, so that it's difficult for them to change sets, as you would. And so one of the tests that we're using, which really is a combination of cortical and cortical basal ganglia tests. So, you can put an electrode in a number of different places and you get responses, and you get some information about how that brain region is responding to it. But we're looking at behaviors in which the patient or the individual has to make a decision about whether they're going to take a reward versus an adverse outcome. And the reward in the cases that we're using in our behavioral paradigm is money. And the aversive outcome is looking at a very upsetting picture. So there are upsetting pictures, there are neutral pictures, et cetera, et cetera. The patient has to decide, and we have normal controls, of course, decide beforehand whether they're going to take the chance, okay, it's a probabilistic test, of looking at the picture or not. And they have a probability of what they know what the reward will be, the probability of the reward, and they know what the probability of them having the yucky picture that they have to look at. So, if you have a low probability of a bad picture and you have a really high probability of reward, so, you get $10 and, it's a good chance you're going to get that. I mean, most people will go for the reward. They'll go for the and the reverse is true. If there's a high probability of a negative outcome, in other words, you are going to see this horrible picture, and there's a low probability of any kind of reward, then you're not going to look at the picture. You're going to choose not to do that. And so then, of course, those are the extremes, but then, of course, you get into the middle where you're balancing it. And so you can imagine that patients, so we haven't broken the blind yet, but there's no doubt there's a population of patients, or we don't know, so there's controls and OCD patients, and we know there's a subset of these individuals, these subjects, that actually clearly will take a very, very low probability of a negative outcome and not look at the picture. They just, even if it's a high probability of reward. The brain regions that we think are very involved in this, in addition to the striatum, are the ventrolateral prefrontal cortex and the anterior cingulate cortex. And I was mentioning this afternoon, we know that there are cells, based on electrophysiology results, in the anterior cingulate that code for both, same cell coding for both reward and negative outcome, and whereas in the VLPFC, they're separate populations of cells. I don't know if that really answered the question, but I think it just kind of painted the view maybe that it's very complex, and we think that, and of course, if you record in the striatum, you'll have, cells that also respond to negative outcomes and positive rewards. So it's a very complex system, but we think that there's a whole network of areas that are important and are involved. And we, in our Conti group, we're focusing on the VLPFC and the anterior cingulate, and of course and other areas too, but of course the striatum is part of that. I'm going to need to quickly get my plug, so go ahead with the next question. I'm listening, but I'm going to lose my connection.
Bom, acho que vocês tiveram uma perspectiva, né, de quanto esse campo é interessante. Então, a possibilidade de se estudar testes neuropsicológicos, mapear em indivíduos, como os indivíduos respondem de forma diferente a esses testes neuropsicológicos, a correlação desses testes psicológicos com vias cerebrais, e a gente pensar em mecanismos, modelos que possam ajudar a entender os sintomas que os nossos pacientes apresentam, né? Eu acho, André, que a gente ficou meio tarde, né? O que vocês acham? Vamos fazer outras vezes, né? Vão ter várias oportunidades. Eu estou pensando, se vocês não se importarem, a gente deixar ela descansar um pouco, ela deve estar muito cansada. Tudo bem? Podemos terminar? Vou agradecer ela e passar a bola para você, então, André. Pode ser?
Ok.
Suzanne, we are very grateful for your presentation. I think you gave us a glance of the potential of this area, and also we are very admired, but we admire a lot this incredible work you have been doing, and I see a lot of perspectives in terms of helping our patients based on the work you have been doing. So congratulations and thanks a lot.
Well, so thank you. I very much enjoyed it, and I have to tell you, Yuri, I won't see you again probably, but I really enjoyed my visit here. It was really fun to meet with all of your group, and I really enjoyed it.
Thank you. Thank you.
Are you going to be at ACMP?
Yes, I will go. I will to ACMP, and I will see you there.
Okay.
I pass to André Lemos to close the session. So André, go ahead.
Sorry.
Pessoal, boa noite, muito obrigado a todos a presença, muito obrigado pela disposição do tempo. O professor Flávio esteve presente aqui conosco o tempo todo. Professor, você quer dar uma palavra?
Eu queria só agradecer a todos, né, pela presença hoje aqui, agradecer a doutora Suzane. Suzane, thank you so much. Fantastic, fantastic, fantastic. Agradecer a Eurípides também, né, por essa oportunidade que nós temos com esse projeto tão bacana como o CISM, de poder envolver tantas pessoas interessantes, que são expoentes dentro das suas áreas e que, através da possibilidade que a tecnologia nos permite, nos traz esse conhecimento tão profundo e tão diverso daquilo que no dia a dia a gente tem verificado. Então, quero agradecer em nome da instituição, da Unifage, da Unimax, a essa parceria que nós temos com o CISM, na pessoa do Eurípides e toda a equipe dele maravilhosa, aos nossos queridos alunos aqui da Unifage, da Unimax, aos alunos das instituições convidadas, né, da USP, da Unifesp, da Federal do Rio Grande do Sul, e a todos que puderam participar aqui essa noite, alunos e professores. E agradecer, claro, ao nosso querido professor André, que foi aí o anfitrião da nossa noite. Obrigado.
Okay, everybody. Bye, Suzanne. Bye,
Suzanne. Thank you very much for sharing your knowledge with us.
Bye-bye.
Bye.
Thank you.
Thank you.